Okay.
Morning.
Morning.
Hi, Steve. Hi,
Morning,
Francis.
Mauricio.
Okay, so all right. Okay, so good morning, everyone, and welcome to our February KCN Crumble Computational Neuroscience Seminar Series. And we have a fantastic speaker today, but I'm going to let Steve do the introduction. So just to mention that these are approximately monthly talks that we've been having since back in 2017. So the Crumble Computational Neuroscience Group, or Hub, as Rishi nicely has on his slide there, we've been doing this, to sort of, give an opportunity for the computational neuroscience community to hear talks in that area. So without further ado, I'm going to pass the mantle over to Steve, who will introduce our speaker for today. So welcome, Rishi, from me.
Okay, good morning, everyone. And today I'm very happy to have with us Rishikesh Narayanan. Rishi was very gracious in serving as the external examiner for one of my students' PhD defenses last month. We were very happy to have him come back this month to give a presentation to all of us. So Dr. Narayanan did his PhD in 2002, finished it at the Indian Institute of Science in Bangalore. He then went on to do postdocs at the National Center for Biological Sciences in Bangalore, and then at the U of Texas in Austin as a Human Frontiers Fellow. He returned to the Indian Institute of Science in 2009 as an assistant professor, setting up his own lab, and has risen through the ranks, becoming full professor in 2021. He was also elected as a fellow of the Indian Academy of Science in 2020 and has a host of other awards. And what really attracted me to his research is his focus on the regulation of excitability and neuron function, network function as well, and dendrites, but specifically the importance of degeneracy, which I think he's going to tell you more about today, but it's something that I think is really important, and his work has really nicely illustrated the importance of that in a host of different areas. So I'm not going to speak anymore because I want to give Dr. Narayanan as much time as possible, but the title of his talk, as you can see, is Plasticity and Computing in the Brain Towards Remedying the Oversimplifications. And so with that, I'm going to pass it over to you, Rishi.
Thank you so much. Thank you so much for having me as part of this seminar series. And thanks, Steve, for for the nice introduction, and thank you, Francis, for the introduction to the introduction, as it were. Okay, so let me start with this. So I was debating between one study talk and a more generalized introduction to what goes on in my lab, and then finally decided to have a more generalized introduction, and Steve agreed with my conclusion and said that it was a good thing to choose this side of it because it was a broader audience, and therefore I'll try and keep it simple and try and provide an overview of what we do, and it will not necessarily be restricted to what we do in our own lab, but it's a more generalized talk, and I'll lace it with what goes on in our lab, basically. And I mean, this is being recorded, and I'll be happy to share the slides as well. Anything works. And if you have any questions, please interrupt me, or you could post questions in the chat box, and Steve would be able to see it. You can interrupt me anytime that you want me to interrupt, basically. All right, so I always place the acknowledgement slides first because these are the people who actually did the work. I don't do any work because once you cross the barrier, as it were, it becomes very difficult to directly contribute to science. And these are the people on the left who contributed to the work that I'm going to be talking about. And funding agencies that have helped us over the years to do what we do in our laboratory, or these organizations that are listed on the right, basically. So these are the people, and these are the funding agencies that helped make this possible. Otherwise, it wouldn't have been possible. I will start from the very beginning. I mean, we can't start neuroscience talk without mentioning Ramon y Cajal, and it was he who proposed the idea of what's called as the law of dynamic polarization, which is something which all of us are familiar with. The basic idea being that the flow of information in a neuronal structure is unidirectional in nature, where the information is received at the dendritic end of it. That's where the electrical signals from other neurons are collected. And then at the cell body, where the integration of all these inputs, the spatiotemporal integration of all these inputs occur at the cell body. And once this input crosses a certain threshold, then at the axonal initial segment, you have an action potential that is generated, which travels through the axon towards going to the botons that are present over here, which make contacts onto other dendrites. Right? So the fundamental idea here is that information flow is unidirectional. There is no flow of information from the cell body into the dendrites. Right? So there is no flow of information from the axon into the cell body again. And you don't have a scenario where there is overlapping information, basically. So this was the law of dynamic polarization, where there is this polarized structure which sustains information transfer in a unidirectional fashion now. Right? So that was the fundamental idea of how a neuron works a long time ago. Based upon this fundamental structure, it was postulated that there will be a bunch of synapses that are present on the dendritic structure. What's shown in green is supposed to be a passive structure, and therefore it's green. And these little spiny structures in blue are these spines, and they receive the information from other neuronal structure, the axons of other neurons. And there is an integration that occurs as a consequence of these inputs that come on to this particular neural dendritic tree. So you have several inputs, X1 to Xn, and there are these different weights assigned to each of these inputs. Each of them contributing not just to the presynaptic properties, the postsynaptic properties of the synapse that is present over here, but also to the kind of attenuation that would be encountered by this particular structure, this particular information that is coming on this particular location, and as it traverses to the cell body. All that is captured by this single number over here. Right? So based upon this idea that there is going to be unidirectional flow of information, you have a summation of all these inputs, and that integration occurs at the cell body. And then eventually, when a certain threshold is crossed, you have a nonlinear function, which is defined by F here, a threshold nonlinearity shown as a sigmoid here, which would elicit or which would fire an action potential once a certain threshold is crossed. Right? So that's why the nonlinearity is represented as this a red structure over here. And eventually, once an action potential is generated, that traverses along this particular structure. Right? So this is the simplest possible model of a single neuron function, and this, as we know, is something that we refer to as a perceptron, basically. So in this simple model, if you look at what can change, right? I mean, if you take a close look at the different components associated with this particular model for a single neuron function, you see that the fundamental place where changes can occur are these weights that are present over here. Right? So that's the place where changes can occur. The other places are, there is a threshold nonlinearity that is present over here, and information flow is kind of unidirectional. And therefore, the idea was that the cellular basis for learning and memory was rooted or was mediated by changes in synaptic structure. Right? So that was based upon this simplistic or this simplified model of what a single neuron does, the only place where changes can occur are the synapses, and therefore it was postulated that synaptic plasticity was what mediated learning and memory in neural structures. So I mean, over the years, there are, there were ideas that were postulated as a consequence of this, and I mean, to summarize what I said so far, I mean, the first part says that, I mean, there are these neurons which are simplified algebraic summation units, which is followed by a threshold nonlinearity. And synapses change in response to learning and form a putative cellular substrate for learning and memory. So that was the fundamental idea of what we called as synaptic learning theory. Right? So over the years, again, several rules have been proposed for how these synapses would learn, what kind of interactions would they have with the pre and post synaptic activity, and whether they would be dependent upon spike timings, whether they would be dependent upon the average activity in the two neurons, whether it is going to be governed by some kind of a threshold, or I mean, is it going to be governed by some kind of winner takes all mechanism that is going to drive synaptic plasticity? That resulted in an array of different rules over the years, and they have been used for explaining different kinds of biological phenomena, ranging from hippocampal synaptic plasticity, how it's dependent upon calcium, or in this case, how orientation and direction selectivity in the cortex kind of emerges during developmental phase and so on and so forth. Right? So over the years, there have been several learning rules that have been proposed towards understanding how learning could be emergent as a consequence of synaptic plasticity, basically. Right? So using synaptic plasticity as a substrate and different kinds of architectures, I mean, you can have a completely feed-forward structure with each of these neurons represented by this simple perceptron over here, or you could have some kind of a recurrent structure where there is much more feedback and feed-forward connection, all sorts of connections, basically, that is present in the structure. Different kinds of networks were proposed, each with different architectures, different algorithms that were used for learning that were created over the years, and there have been several different applications. Right? I mean, there is no question about the utility of these networks, which have been constructed with this simplified perceptron-like neuronal model, and how it has been used for several purposes. I mean, even within the neuroscience community, Deep Lab Cut, which is a software package for animal pose estimation and behavioral analysis, is very popular, and there is no question, as I mentioned, as to the utility or the success of these algorithms, these networks, from the perspective of how exactly they can be used for analyzing neural data, or getting insights about neural data, basically. So, but the question that we are going to be looking at is not necessarily the question of how these networks are useful from the perspective of analyzing neural data, but from the perspective of whether this oversimplification of a neural structure and this idea that it's only synaptic changes that mediate learning and memory, is it something that is representative of biological system? Right? So is such a network which is constructed of such simple neuronal models reflective of or a realistic model of what exactly happens in biological systems, the nervous system, and so on. So that's the fundamental question that we are going to be addressing here, right? I mean, that's the question that we are going to be asking. Right? So in this narrative of having a simplified neural structure with synaptic learning as the fundamental substrate for learning, it turned out that we were driven by several different oversimplifications. What I'm going to do in today's talk is I'm going to list 5 of them. I'm going to pick 5 of them. Those 5 are, as I mentioned in the abstract, also are just a subset of the oversimplifications that are out there. I'll list some others at the end of it in the last slide, but I'm going to pick up 5 different oversimplifications that we came to know afterwards, beyond what Ramonica Hall and beyond what those algorithms mentioned as a consequence of research, biological research over the past 2 or 3 decades, or mostly 2 or 3 decades. And we came to understand that there are these different oversimplifications that are present in this narrative. I'm going to pick up 5 of them. I'm going to tell about the kind of work that has been going on in not just my laboratory, but in other places also, in terms of how to remedy this, how to make sure that these oversimplification don't stifle a wholesome understanding of the brain. The idea is that, the brain is a complex system, basically, right? So in understanding it, we do come up with certain parsimonious explanations, certain oversimplifications, but it is also critical that we don't make this, we don't allow these oversimplifications to block our understanding of the whole system as a whole, the system as a whole, and understand it for what it is, rather than understanding it for the model that we are presenting it to be happen. Right. So the first oversimplification that I'm going to be talking about is the very fundamental assumption or the idea that neurons are simple algebraic summation unit with a threshold nonlinearity. Right. So that's the first oversimplification. So the idea at that point of time was that the dendritic structure was passive in nature, which means that it doesn't have any quote unquote nonlinearity that is present over there, and it just summates the kind of excitatory and inhibitory inputs that come onto the structure and sends it to the cell body where there is a threshold nonlinearity that is sitting over there, which converts it into an action potential. And so, fortunately or unfortunately, research in the 90s and early 2000s, where people actually patched onto these dendrites using cell attached recordings and asked the question of whether these dendritic structures, do they have quote unquote the nonlinearity? I'll come to what exactly that nonlinearity is. And they found that the nonlinearity was not something which was limited only to the cell body, but it was prevalent throughout the entire dendritic structure. Right. So where does this nonlinearity come from? Right. I mean, as I mentioned, it's the ability of the cell body or the axosomatic compartment to elicit an action potential that mediates this nonlinearity. So here is an example from a hippocampal pyramidal neuron, CA1 pyramidal neuron, where you're injecting a 250 picoampere current injection, a pulse current injection, and you see the cell charging up. And once it crosses a certain threshold voltage, it elicits an action potential and it comes back to where it was. So this is the integration part of it, which is kind of represented by this large sigma over here. And then there is this fire part of it where you cross a certain threshold and that elicits an action potential. And these action potential, as Hodgkin and Huxley taught us a long time ago, is mediated by the ion channels that express on the membrane. So there are these transmembrane proteins which are selective to certain kinds of ions. And there are sodium and potassium channels that are present on the membrane. And the interaction between them, I mean, these are functionally segregated molecules as they were. I mean, one is a sodium channel and the other is a potassium channel. And the interaction between the two of these functionally segregated subsystems gives rise to a functionally unified outcome, which is the action potential at the cellular level. And that is what, I mean, the presence of this ion channel is what mediates the nonlinearity. And as it turns out, the nonlinearity is specifically consequent to the voltage dependent properties of these ion channels. So if you put voltage on the x-axis and if you put the open probability of these ion channels on the y-axis, you would see some kind of a sigmoid, which is what mediates this nonlinearity. And the point at which the sodium channel is open is what contributes to the threshold that is present in these neuronal structures. Right. So now the question is, if we have found out that these ion channels are the ones that are mediating this nonlinearity over here in this neuron, then the question comes to whether these ion channels are present only in the axosomatic compartment, and therefore you are allowed to make this approximation that the rest of it is just passively integrating, or are they present throughout the entire dendritic structure, where you would have to now account for the presence of these nonlinearities even in the dendritic structure and cannot make this assumption over here about a simple summation that is present over there. Right? So as I mentioned, people in the 90s patched onto neuronal structures at different locations, and they found that the ion channels are present not just at the cell body or at the axonal initial segment, they are present throughout the entire dendritic structure. Right? So, I mean, you have some of these ion channels like the A-type potassium or the HCN channels. These ion channels are present at higher densities in the dendrites in hippocampal and cortical pyramidal neurons compared to what you find at cell bodies, basically. And if you look at the voltage dependent activation properties, they also exhibit these nonlinearities, which means that the nonlinearities that we thought were present only in the axosomatic compartment are not confined only to the axosomatic compartment, but they are present throughout the entire dendritic structure. Right? So these dendrites which express these active ion channels, when I say active, I mean voltage-gated, calcium-gated ion channels, are what are called as active dendrites. And it turns out that the presence of these active dendrites can confer upon neurons computational powers which would be impossible to think of if you consider just a passive dendritic structure. Like say, for instance, the fundamental assumption of the law of dynamic polarization that there is unidirectional flow of information from the dendrites to the cell body to the axon is gone because you have action potentials propagating back into the dendrites because of the ability of because of the presence of sodium channels along the dendritic structure over here. Right? So, and the fundamental assumption that action potentials are all or none signals that it mean either they are this 120 millivolt signal or are they zero. That also went out of the window because if you record these back propagating action potentials and measure their amplitudes, you would see that they are graded in nature. If you go down to something like 300 microns in a hippocampal pyramidal neuron, you would see that the amplitude of these action potentials are on the order of 5 millivolts. Right? I mean, they look like EPSPs, except that the kinetics is much more faster. And the idea that if there are sodium channels that are present in the dendritic structure, the idea that action potentials can be initiated only at the cell body also goes out of the window. It fundamentally means that you can initiate spikes in the dendrite. And they are mediated not just by sodium channels that are present over there. There are calcium spikes that can occur in the dendrites. There are NMDA receptor dependent spikes that are occurring in the dendritic location. So the whole idea of having a simple integrator to model the entire dendritic tree, which kind of feeds into the axosomatic compartment for generating an action potential, went out of the window as a consequence of the presence of ion channels along the dendritic structure, right? So, and they don't even act as simple integrate and fire low pass units. When you have an integrator, it acts as a low pass structure, but you would have as a consequence of certain fancy ion channels like the HCN or the M-type potassium, you would have band pass filtering introduced at different locations. And I mean, for those of you who are used to think of signal processing from a kind of filter bank kind of an approach, you can think of each and every location in the dendritic tree responding to different frequencies, basically. So you can have different densities of HCN channels here versus here, and therefore the filtering capabilities of this dendrite over here and the one here will be very different, and the frequencies that they will let pass are also going to be very different as a consequence of what is present at these locations. Right? So you have this this powerful machinery that comes into picture as a consequence of the expression of different kinds of ion channels in the dendrites. So again, over the years, we have also been looking at the implications of what active dendrites could do, or what are the functions of active dendrites at different scales of analysis. Right? I mean, at the network scale, we have been looking at if there are active dendrites, then how do they contribute to local field potentials? Like say, for instance, I mean, at one point of time, it was assumed that you have, one is the synapses that are present on the dendritic structure, which means that if you filter out the action potentials in the extracellular potential, then you would have pure synaptic inputs that are coming onto that structure that will reflect in the LFP. But with the presence of ion channels in the dendrites, it becomes very difficult to say that it is purely synaptic structure, but you will have to account for these ion channels that are present over there. Right? So we also looked at the impact of glia transmission. I'll come to this at the end when we talk about glia. But I mean, this also has a strong interactions with the active dendrites. Without active dendrites, there would be just large synaptic inputs, but with the presence of active dendritic structures, it becomes a nice and beautiful plateau and gives much more signal processing capabilities, basically. We have been also looking at frequency selectivity in the spike triggered average, which is close to Steve's heart, and coincidence detection class 1, 2, 3 neurons. And we think of class 1, 2, 3 as not properties of neurons, but as properties of compartments, because you have this differential expression of different kinds of ion channels in different compartments, and therefore, say for instance, the distal dendritic region could be class 2, 3, and the soma could be class 1 as a consequence of a higher density of HCN channels in the dendrite, basically. Right. So, and we have also been looking at the impact of distributed synapses and how active dendrites and dendritic spikes could be contributing to spatial information transfer in these structures. At the molecular level, we have been looking at how the presence of this active dendritic structure could allow for calcium wave propagation, as well as how signaling microdomains could be propagating in the dendritic structure as a consequence of presence of these different ion channels that are present over there. Right? So over the years, we have been looking at the implications for the presence of active dendrites across different scales of analysis. And I mean, to summarize this oversimplification, that a single neuron is a simple integrate and fire structure. The remedy for that is, or the actual observation that we see in biological neurons, is that a single neuron is a complex computational machine with active dendrites, which is why I've made all of this red, sustaining bidirectional information flow. Information flow is not unidirectional anymore. You can have backpropagating action potential, you can have dendritic spikes, you can have bandpass filtering, and so on and so forth. There are a few reviews that I have listed here, which is this is from Yota's lab. And there is this wonderful paper, which I strongly recommend for those of you who are used to looking at neurons as perceptrons. So this is a paper where they show that you need an entire deep artificial neural network of 7 layers for you to be able to characterize a single cortical neuron's input-output characteristic. Right? So I mean, a single cortical neuron is not a perceptron, but a deep artificial neural network, which is made up of several different layers of perceptron, basically. And that gives you an idea of the kind of complexity that we are dealing with in terms of a single neuron and what it can do and what actually does. So it's something that you have to keep in mind, that this is not representative of what an actual biological neuron does. It's something which is much more complex, much more powerful. And there have been several laboratories that have been working on this. Some of these reviews will give you an understanding of what goes on. Right. All right. So that's the first oversimplification of 5. The second oversimplification that comes into picture when we think about artificial neurons, artificial neural networks, or when we think about even neurons in general, is that we think of neural circuits being made of repeating homogeneous computational unit. There is no difference between neuron 1 and neuron 2, and they're all exactly the same. They all integrate and fire, and they just talk to each other, and they produce a certain output. It's just the connectivity patterns that produce this outcome that we see in terms of final outcomes, right? So, but the reality, based upon, again, experimental data from several different laboratories, is that neural circuit heterogeneity is manifest in several different forms. Intrinsic heterogeneities, which means if you inject the same current to different neurons, even adjacent to each other, they are going to respond very differently. synaptic connectivity, structural properties in terms of the morphology of the neuron and the spine structures and so on and so forth. And when I say afferent heterogeneities, I mean the connectivity from another area to this particular structure and where exactly. I mean, it's not like all the fibers from the afferent structure are going to be making contact with every single thing that is present over here. There is going to be a distribution. There is going to be heterogeneity over there also. That's something which is seen across neurons. Right? So these are some examples. So these are examples from the same brain region, the dentate gyrus. These are all granule cells. And you can see the arborization, which is very different across these different structures. These are all CA1 pyramidal neurons. And again, you can see the arborization to be very, very different. Even within the same cell type in the same brain region, you would have very distinct morphologies that are present. So this is, recording from Poonam. So these are nice and beautiful dentate gyrus granule cells, and she's recording from one single neuron over here, and you see that there is a 250 picoampere current injection, and this elicits a firing pattern, and you have a firing frequency of 26 hertz. But if you now pass on to other neurons that are present either in the same slice or other slice of the same brain region, you would see that there is a distribution of how exactly this firing frequency manifests for the same 250 picoampere current injection. It's not like all the neurons that are present over there exactly give you 26 hertz of firing frequency, but some of them don't fire at all. Some of them give you close to 40 hertz of firing frequency for the same synaptic input, for the same, in this case, the current injection, and by extrapolation, for the same synaptic input, you would have very different kind of firing patterns or firing rate that's observed in these systems, basically. Right? So there is considerable heterogeneity in terms of the intrinsic physiology. Yeah. And as I mentioned, there are several different local heterogeneities. The structural properties could be different. The intrinsic properties, by which I mean the input resistance, the firing rate, and so on and so forth. And in terms of afferent heterogeneities, these are the fibers that are coming from another brain region. It's not like all the neurons are getting identical inputs from the previous, the projecting brain region, but you would have very different set of inputs making contacts. There could be some overlap, but there is heterogeneities even in terms of the afferent connectivity, basically. Right? So you have these local and afferent heterogeneities that are present in a given neural structure. Right? So you may ask, okay, you may just tell me that, okay, they are present. Why don't we just ignore them and move on? Why do we have to even account for these, basically? It turns out there are some important implications for the presence of these heterogeneities. I'll give you two examples. In one where these heterogeneities seem to be fundamental in terms of executing a certain function, and another where the presence of these heterogeneities seem to impair a certain function, but can be restored if you take an additional biological reality into account, basically. So one of the important postulated functions of the dentate gyrus, which is the input gate to the hippocampus proper, is what's known as decorrelation. You can think of it as pattern decorrelation or pattern separation. So the idea is that you want to discriminate between inputs that are very similar to each other. Even if the inputs are similar to each other, you want to have the outputs to be very distinct from each other. You want to distinguish, for instance, between these two phases. And even though they are similar, you are able to distinguish between them and say that one is really different from the other. So one of the postulated functions from Marr's time, David Marr's time, has been pattern decorrelation or pattern separation of the dentate gyrus, basically. Right? So it turns out that if you have, the idea that the presence of intrinsic heterogeneities could separate very similar patterns or even identical inputs is relatively old. Right? So let's say that you have identical inputs coming onto two different neurons, and they have very different excitability. Right? Which means that their firing rates are going to be, the input that is required for eliciting an action potential is going to be very different. One has a very low threshold, the other has a very high threshold, let's say. So the one that has lower threshold will fire more than the one that is having a higher threshold. And that itself introduces a certain amount of decorrelation between the inputs, basically, even for identical inputs. So here is a scenario where, where different kinds of heterogeneities were taken into account. So this is a population. This is the outcome of a network which is completely homogeneous, where there are exactly the same set of neurons that are present. And there is only synaptic heterogeneity. But if you look at correlation with identical kind of inputs, you see that, it's close to 1, basically. Right? So on the other hand, if you have a certain amount of intrinsic heterogeneity, which is to say that each and every neuron that is present in this network has different excitability properties, different integration properties. So you observe that the presence of just the intrinsic heterogeneities brings about a certain amount of decorrelation. So, all that you have to know here is if it is lesser than 1, it basically, there is a bunch of lesser than 1 population over here, and therefore this brings you a certain amount of decorrelation, just the presence of intrinsic heterogeneity. Now you could add, structural heterogeneities on top of this. Where does the structural heterogeneities come? The dentate gyrus is one of 2 brain regions where adult neurogenesis occurs. And therefore, you have these immature neurons, which are smaller in size, and they mature to become larger. And that gives you the structural heterogeneities. And you see that that also brings about a certain amount of heterogeneity, certain amount of decorrelation, basically. So in this case, the heterogeneities themselves are mediating a certain function, which is decorrelating the inputs. Even if the inputs are very similar to each other, they're able to distinguish between them, right? So, but here the idea was to ask questions about which of the different heterogeneities have the prominent role or the most dominant role in terms of giving out, or mediating decorrelation. And it turned out that if you have these afferent heterogeneities where the inputs themselves onto these different neurons are different, that gives you the maximum amount of decorrelation. And the presence of that, these intrinsic and synaptic structural heterogeneities don't do much, basically. So here is one case where the presence of heterogeneities themselves mediates an important function associated with this. It also turns out that the presence of these heterogeneities could bestow upon these networks a certain functional resilience to perturbations, basically. So here, perturbations were introduced by taking out individual ion channels in the network. So this was a conductance-based network model. And here, in the presence of, in the absence of structural heterogeneities, you see the amount of perturbations to be large. Here, the output is measured in terms of the decorrelation that can be achieved. Here you see that as a consequence of the perturbation, you see a huge change that is present between the perturbed one and the baseline, which is shown as 0. And on the other hand, another network where there were structural heterogeneities present, you observe that the amount of changes that you observe in the output correlation has reduced as a consequence of this, and therefore the presence of these heterogeneities also introduce functional resilience to these neural circuits as a consequence of that presence, basically. Right? So and if you look at heterogeneities, they are not limited only to basic properties. They are also, manifest in terms of the amount of plasticity that is introduced by any protocol. It doesn't matter what LTP protocol you are used to, what plasticity protocol you are used to, what intrinsic plasticity protocol you are used to, there is always a heterogeneity that is observed. Typically, you would observe LTP experiments being reported like this, where you have a baseline, you give the protocol, and you have, under baseline conditions, you have around 250% LTP. And under, when you block certain receptors, let's say NMDA receptor or whatever that is, you see that there is a reduction in the amount of plasticity, basically. So this is how typically data is shown, and you would typically compare these two ends and say that, oh, this is significantly different, and therefore it is blocked. Well, if you look at the actual experiments, so this is made up of several different experiments across several slides, several neurons. If you look at the actual setup in terms of, or the actual outcome in terms of how exactly the amount of LTP manifests. So this is the control data. So you see that there were several neurons where there was not much of LTP as a consequence of bringing about that protocol. And this 250 is a consequence of some of these points that are present over here, basically. Right? So on the other hand, when you actually block the NMDA receptor or whatever that molecule is, you see that it's not like it was a straight line where you have everything lining up over here. You have some of them showing plasticity, which is comparable. If you look at the amount of plasticity that you observe here and the amount of plasticity you observe in the control scenario, you see that there are several experiments where they are comparable, actually. Right? So it's the usual way of interpreting that is you take the average, you take the SCM, you do a t-test or something like that and compare it and show that it is p is less than 0.001. And we will see in a minute about what we call as people have called as degeneracy, where there are several components that contribute to the amount of calcium that comes inside. And therefore, there are certain cells where the blockade is not necessarily blocking the plasticity. And therefore, it's very important to show this kind of a plot. Not many laboratories show these kind of cumulative plots showing the actual distribution of data. But it's it's very important that you don't just show this, but also show this, basically, so that people get an idea of what kind of distribution is present over there. So in this study, we looked at what exactly is the impact of the different heterogeneities that I talked about, synaptic, intrinsic, and structural, on the plasticity profiles themselves. This is a BCM-like profile, and you observe that each of them can bring about heterogeneities in the plasticity that is induced, basically. So that is just to show you that there are these heterogeneities that are present. And I showed you one example where that was helpful in terms of providing the kind of outcome that, providing the functional outcome that we were looking for. And here I'm going to give you another example where the presence of heterogeneities kind of hampers the function. And if we introduce another biologically relevant thing over there, we will restore it, as it were. So these are the grid cells. As all of us know. So you have, so this is an arena that is present over here. The black lines are an animal running around in this arena, and the red dots over here are spikes that are recorded from a single medial entorhinal cortical cell. Right? If you now take the firing rate of this cell and plot it, you see that it forms this hexagonal pattern or triangular pattern, which is why these cells are called as grid cells, basically. And ever since these were discovered, there have been several models that have been used. One of the most successful class of models that have been used to explain this are the continuous attractor models for grid cells from Burak and Feet. So they showed that you can have a certain kind of connectivity, and you will be able to replicate this kind of a patterned activity that is present in these structures as a consequence of the connectivity and how they talk to each other, basically. Right? But most of the models that are used for explaining grid cells or the network models, they don't have any kind of heterogeneities, basically. They are made of homogeneous neurons, right? And they talk to each other. So here what Devyant did was, so he introduced heterogeneities one by one, the different heterogeneities that I talked about in terms of how different individual neurons could be, what kind of inputs come on to each and every neuron, what kind of synaptic properties are there, and then all of them together. So here you find that if you keep on increasing the degree of heterogeneity, you see that at this degree, you still see some kind of pattern that is present over here. But at this degree of heterogeneity, you see that the pattern of activity, the grid pattern activity, is completely gone, basically. Right? So you have a disruption of this pattern firing as a consequence of introducing these heterogeneities. And what Devyant asked was, what exactly is mediating these this disruption, right? So So he measured activity as the activity of these neurons under various different levels of heterogeneities, and he found that the perturbation to activity was predominantly in the lower frequencies. Right? So when this is the variance of differences between what he observed in the homogeneous network versus what he observed in this heterogeneous network with different degrees. And you see that the amount of variance in the differences is increasing with the degree of heterogeneity, and it's kind of, limited to the lower frequency. So the question was, if we now suppress this lower frequency perturbations, which is consequent to the introduction of heterogeneities, would we be able to get back grid pattern firing? So that's the fundamental question that he asked. And it turns out that's a very good way by which we could actually suppress lower frequency inputs in a very biologically relevant manner. So there are these ion channels that are present, which are called as either resonating conductances or phenomenological inductances, or pacemaker channel. The HCN is one example of that. The M-type potassium is another. If you have these kinds of ion channels, then neurons don't behave like low-pass filters. So if it were a low-pass filter, you would have it going and converging over here, but act as band-pass filter, and it is achieved by suppressing the lower frequency. So that's what these resonating conductances do. So the question was, if we now make these integrator neurons, which is what typical models of grid cells assume these neurons to be, but if you look at entorhinal cortical interneurons and the medial entorhinal cortical interneurons, as well as the principal neurons, you would see that they express resonance. And therefore, Divyan said, okay, let me introduce resonance through two different ways. One by some kind of a brute force suppression of lower frequency by introducing an additional high pass filter. Another that mimics the resonating properties by introducing a negative feedback loop, which is slow onto this particular structure. And both of them gave him resonance. Now he replaced all the neurons in the network by resonators rather than having them as integrators. These are class 2 excitability, if you them. So in the integrator, we observed that there was a disruption in the grid cell firing pattern. But if you replace the neurons by resonators, where we are now suppressing the perturbations that are observed as a consequence of heterogeneities, you see that you still have this grid pattern firing coming forth as a consequence of that. Right. So the resonance was something that was able to stabilize these heterogeneous grid cell networks, and that's another thing. So again, over the years, we have been looking at the implications of neural circuit heterogeneities in different brain regions. Our focus has always been in the hippocampal formation, which covers the entorhinal cortex, and we have been looking at the implications of this is the decorrelation work and the plasticity work, and this is the grid pattern firing network that I talked about. So fundamentally, there are different kinds of heterogeneities, and it's important to not consider the neural circuits as repeating homogeneous, made of repeating homogeneous computational units. There are also other studies which talk about how neural heterogeneity can promote robust learning. This is from Dan Goodman's lab. And here they say that learning with heterogeneity was more stable and robust, particularly for tasks with that rich temporal structure. Right. So there have been several other laboratories which have been looking at the implications of heterogeneities. And it's important to remember that experimental analysis and computational models should account for all the neural circuit heterogeneities rather than treating everything as some kind of homogeneous repeating units. Right. All right. So the third oversimplification that I want to talk about is the idea that learning and memory in biological system is accomplished exclusively through synaptic changes. This started much before Hebb. One could call Hebbian plasticity as Andersonian plasticity, because if you look at Hebb's rule, you see versions of it in James Anderson's books a long time ago. So the idea that it is synapses where changes occur has been around for a while, and that's what kind of drove us to this idea that I mean, learning and memory is kind of accomplished exclusively through synaptic changes. But in reality, thanks to our ability to measure plasticity, not just in synaptic changes, but also in things that are mediated by ion channels, like say, for instance, I mean, all the things that I talked about, back propagating action potential, input resistance, resonance frequency, each and every component that is part of a neuron can undergo plasticity. And that is very nicely summarized by this review, which came a long time ago by David Linden and Kim. So please read this, and it will actually show you. Now there are several more lines of evidence to show that plasticity is indeed ubiquitous and not something which is limited only to synaptic changes. Right? So and it's not like it was something which was known only later on. The fact that there are intrinsic changes in neurons that accompany synaptic plasticity, that was shown in the first ever paper that showed synaptic plasticity electrophysiologically. Right? So this is the famous Bliss and Lomo paper, J Physiology, 1973. And here, I mean, this is the abstract. It's not hidden in some obscure part of the paper. It's in the abstract itself. So the results suggest that two independent mechanisms are responsible for long-lasting potentiation. One, an increase in the efficiency of synaptic transmission at the perforant path synapses. This is what the literature ran with and called that as LTP. And two, an increase in the excitability of the granule cell population. And if you read through the paper carefully, you would see that there are experiments where they don't observe A, but they do observe B. You would see that there is an increase in excitability, but no change in synaptic strength, basically. And they even if you go into the details, they would say that, oh, there could be changes in intrinsic factors, such as those which determine threshold, like say, for instance, sodium channels and stuff like that. So it's not something which is novel or new. It has been there ever since the first ever demonstration of synaptic plasticity. But it is just that because of the Hebbian bias that we had, we just chose to run with one of them rather than taking both of them and treating them as equal. I mean, it's much more than these two. As I said, plasticity is ubiquitous. So instead of taking the two of them and running with both of them, we just chose synaptic plasticity and said that everything is mediated by synaptic changes, basically. Right? So in today's world, I mean, this is our viewpoint, our synthesis of how to account for this ubiquitous plasticity. How is that you get stable continual learning if plasticity is kind of ubiquitous? If everything can change, and if everything can undergo plasticity as a consequence of activity that is coming from the external world, how is that the biological system is capable of maintaining stability and continual learning? So we think of it as strong plasticity manifolds where there are strong constraints on what things can change together. It's not like any arbitrary thing can change in any arbitrary direction. If component A goes up, then component B also has to go up, and component C also has to come down, basically. So that imposes a low-dimensional manifold within which this plasticity exists. And that is what we think of how the biological system is capable of doing or capable of performing stable continual learning in spite of or as a consequence of ubiquitous plasticity. Right? So there are structural changes, there are synaptic changes, there are intrinsic changes, there is global intrinsic changes, there is global synaptic scaling where every single AMP receptor in the neuron undergoes plasticity either up or down. So there are changes in pretty much every component in every possible spatial combination. And it's important to keep this in mind and not just think of synaptic plasticity, basically. Right? So again, over the years, we have been thinking, I mean, working on this has been limited to the hippocampus proper. We have been looking at interactions between synaptic and intrinsic plasticity, and both experimentally and theoretically, and how exactly different kinds of activity patterns give about changes in these neurons over the years, basically. So there are several very good reviews out there which talk about ubiquitous plasticity, intrinsic plasticity, And this is, much. This, you look at the year over here, it's not something which is new, but again, people don't think of plasticity to be something which is ubiquitous. They always think of only synaptic changes, and a bunch of reviews from our own lab are also there in here, basically. Right? So that's the third oversimplification, that plasticity is not something which is limited only to synaptic changes, but is something that extends to other components as well. The fourth oversimplification, which is one of our, all of these are strong research themes in our lab, but this is probably the strongest research theme, which Steve also alluded at the beginning of the talk. I come from an engineering background, so for me to think of even multiple solutions is kind of beyond my thinking capabilities. Therefore, I used to think of biological solutions also to be kind of unique. There is one solution that is present over there, and we are supposed to find that solution, right? So that is how I used to think of biology when I came in first into biology. That's because there is a certain precision in terms of how we design things in engineering. Even though it's not like there is that precision is what kind of guided my thinking, as it were, right? So it turns out there is not one solution to any functional outcome, functional output, but there are several possible routes, and that is codified in this word degeneracy, which is very well elucidated in a review that will come in a minute, basically. Right? So it turns out that biological systems express degeneracy across all scales. So what is degeneracy? So this is Gerald Edelman. He got his Nobel in physiology and medicine in 1972 for the chemical structure of antibodies. Right? So nothing related to what we are talking about as of now. He's not here for his Nobel. He's here because he wrote this very fundamental, insightful, and one of the most important reviews that I ever read. It changed my perspective of how I think about biology as a whole. And I strongly recommend to any brand of biologists, it doesn't matter whether they work on the genetic end of it, or at the evolutionary end of it, or at the cellular end of it, systems end of it, doesn't matter which end of it you work on. Please read this review, and it will definitely change the way you look at biology, basically. Right? So degeneracy has nothing to do with neurodegeneration or degenerates or any of things. It's a fundamental biological concept. It's the ability of the elements that are structurally different to perform the same function or yield the same output. So that's the definition of degeneracy. It is not redundancy. Many people confuse degeneracy with redundancy. Redundancy is a scenario where the same function is performed by identical elements. Like say, for instance, I replace this computer by another and give the same talk. That would be redundancy. On the other hand, if I fly from here to Toronto and give this talk with the chalk and a board, that would be degeneracy, as it were, right? Different structural components, same lecture, same things conveyed, right? So you have very distinct structural components that mediate the same functional outcome. So that's what degeneracy is. It involves different structural components and may yield same or different function depending upon the context in which it's expressed. So this is a table from the Edelman Galley paper. I'm not going to read it out, but if you look at the scales of analysis, at this end of it, you see the genetic code where many different nucleotide sequences encode a polypeptide. And at this end of it, you have an example from behavioral side of it, interanimal communication. There are large and sometimes nearly infinite number of ways to transmit the same message, a situation that is most obvious in language. As said, irrespective of what your brand of biology is, please read through this paper. It's a short paper, shouldn't take much time, but it'll change the way you think about biology and how things can be implemented. Right. So I'll just give you, I don't want to go into details because I think we are already running out of time. So this is one of the first papers where we showed that in spite of the presence of several different functional map, as we call it, where the back propagating action potential amplitude, for instance, reduces with distance, the resonance frequency increases with distance. In spite of all these functional maps being present on the same exact morphology, mediated by various different ion channels that are expressed on this particular structure, you can still have very different sets of ion channels giving you all of these different functional maps on the exact same morphology, basically, because of the kind of interactions that we have between these ion channels and how they contribute to the outcomes. So this is not a electronically compact neuron. This is a CA1 pyramidal neuron, and it is it manifests attenuation. The lambda is on the order of 150 microns, and in spite of that, you see that you can get all these functional, or as a consequence of that, you have all these functional maps obtained with various different combinations of neuron. We have extended these ideas to place cell the firing rate code, where these are hippocampal place cells, and you get exactly the same kind of tuning curves on the one-dimensional spatial axis. That's what is on the x-axis. There's a firing rate. And if you look at the kind of ion channel combinations that give you this outcome, they are very, very different. The same thing holds for phase code as well. This is phase precision, space on the x-axis, and the phase of action potential firing with reference to the theta frequency oscillation on the y-axis. Very similar phase code, but if you look at the models that yielded this this outcome, they're very different in terms of the set of ion channels have contributed to it, basically. So over the years, we have been, as I said, it's one of the strongest themes of research that have been running for almost more than a decade, actually. The first paper came in 2012, and it's still going strong in terms of the fundamental properties, which are in black. The plasticity profiles, you can get exact same plasticity profiles with very different ion channels, very different synaptic properties. Encoding characteristics like firing rate code or like phase code, and we have been looking again at the hippocampal formation, the suprachiasmatic nucleus, where we looked at circadian rhythms in firing rate properties is just the only one that is out of the hippocampal formation. But the rest of it is within the hippocampal formation, and we have been looking at various different aspects, trying to take it forward, not just from the perspective of basic physiological properties, but even in terms of encoding, in terms of plasticity properties, in terms of neurons which are not necessarily electronically compact. We've been trying to take it forward in every possible direction in various different ways, basically. So this is a review that we wrote in 2019, which gives you a broad idea of this work, and these two are more recent ones from different perspectives. This is from the learning perspective. This is from, efficient information coding perspective. Look at degeneracy in different aspects. So please take a look at it. So I don't want to read the whole thing. This is the modified from the original one, which says nothing in evolution, nothing in biology makes sense except in the light of evolution. Because of the strong links between degeneracy and evolution, this is kind of a corollary to that. Nothing in physiology makes sense except in the light of degeneracy. And we think of complexity as something that is necessary, that is essential, and it's evolution's ingenious solution to achieve robust biological function. We don't think of it as curse of dimensionality or something that you have to be afraid of, but it is part of why biological systems thrive, why they are robust. So we think of complexity as something that aids it. So the last one, which should be brief, is about the glial cells. So far we have been talking about neurons, which are one cell type in the brain, and glial cells get their unfortunate name because people thought that they were just holding neurons in place so that they could do that function. So if you look at the active dendritic literature, we talked about passive dendrites and how people came to know that, oh, there are, ion channels that are present and therefore they're active. Similarly, you would see that there's a parallel literature which talks about glial cells as glue, and people started realizing that these cells can have calcium waves sustained in them. They can communicate with each other across the entire network. And that brought about the idea of active glial communications, and they can release gliotransmitters. So there is a parallel literature in this study. What in the study that I'm going to about show, we kind of linked those two parallel literatures and asked questions about whether they talk to each other. So it turns out again, not just from our own laboratories work, but from several other laboratories, it's now known that glial cells are actively involved in information processing, plasticity, and learning. It's not something that is just glue, just there to hold the neurons in place, basically. Right? So this is work by Sufyan, and what he did was he characterized the impact of gliotransmission on the active dendritic structures. He patched onto different locations along the hippocampal pyramidal neuron somatic dendritic axis and asked questions about how exactly gliotransmission manifests. So far, before this, it was known that there were these slow inward currents that manifest as a consequence of gliotransmitters. So he actually asked what exactly do they manifest as in the dendrite when you have these gliotransmission occurring in the proximal and the distal astrocytes that are present over there. Right. So the protocol here is as follows. He patches onto a neuron and measures gliotransmission, these signals. Here synaptic transmission is completely blocked. There is no sodium channels that are present. The AMP receptors are blocked. So you have this period where he measures this. And then he patches onto a proximal astrocyte with the IP3 in the pipette, which activates the IP3 receptors. There will be calcium release, and as a consequence of that, there will be release of gliotransmitters, which would make these strong, yield this strong, high amplitude, slow inward currents, which are mediated by slow NMDA receptors, which he found results in these platopotentials. You can look at the time scale. It's like 200, 300 milliseconds, and you have the amplitude to be on the order of 20 millivolts, 10, 20 millivolts, basically. So these are huge signals, and that's what comes as a consequence of gliotransmission. He looked at the impact of the prominent A-type potassium channels and HCN channels which are present in the dendrites, and found that they do affect the amplitude and the detectable frequency of this. And with additional simulations, he showed that these platopotentials can indeed bring about dendritic spikes, and some of them can convert into full-blown action potentials. Right? So these were, and he also showed with specific NMD receptor blockers that these are mediated by NR2B and NR2D receptors, which are extrasynaptically present, basically. Right? So you can see that these large amplitude events can actually generate dendritic spikes, and which are regulated by the presence of these dendritic ion channels, and that's contributing to how and there have been several lines of evidence that these strong platopotentials can indeed result in plasticity, in synaptic plasticity and other forms of plasticity because of the amount of calcium that they bring in. Here, this gives a line of evidence that there are strong interactions between the glial cells and neurons, neuronal dendrites, and the active components of both of them in terms of mediating or regulating information processing. So again, there are several reviews. This is just one subset of it, which are out there, and there have been several others which have been coming along, which talk about the important role of glial cells in information processing, in plasticity, in circadian behavior. So here we kind of mapped the parallel literatures in the glial cells and the passive dynamic structures and showed that they do interact with each other. Basically, this is also a review. So it's important to keep in mind that the oversimplified idea that neurons are the only ones which are mediating information processing, and glial cells are just there to hold them in place. Is the old one and something that has to be revised to account for the fact that these cells do contribute actively in information processing, plasticity, as well as learning. Right. So to summarize the 5 different oversimplifications, so in the first one we talked about the perceptron, and now you will have to revise that and say that a single neuron is a complex computational machine with active dendrites that can sustain bidirectional information flow. The second one, we talked about the oversimplification that neurons are repeating homogeneous units. You'll have to replace that by the idea that neural circuits manifest different forms of heterogeneities, and we saw some functional implications for the presence of these heterogeneities, and they can't just be ignored. Plasticity is not something which is limited only to synaptic changes, but also to other components. It's ubiquitous. And it's not that there is one solution to a given problem. There are several different solutions to a given problem, several different structural components, and their combinations could give you the exact same physiological outcome. Just because you see that the physiological outcome is exactly the same doesn't mean that the components that are mediating this particular functional outcome are exactly the same. There are a wide variety of components that can come together to this. This is a characteristic of complex systems, and this is something that is ubiquitous across all biological systems and has to be accounted. I mean, I tell this to my students over and over again. It is not just sufficient to know that there is degeneracy. It is important to account for that in your experimental design, in your analysis, and in your modeling. Not just say that, yeah, I mean, there is degeneracy, you can get it through multiple different ways. That's not sufficient anymore. It's important to actually account for that in your, starting from the time where you are designing your experiments, from the time you are, I mean, during the time that you are implementing it, during the time you are interpreting your results. It's very important to account for the presence of degeneracy in the system at each and every scale, basically. Right? So, and it's not like glial cells are just glues. They are actively involved in information processing, plasticity, and learning. And there are several other oversimplifications which need to be accounted for. I'm not going to go into the details of any of them. I just put some, again, even including these are just a subset of the oversimplifications that are out there, and it's important to excuse me, it's important to ensure that we don't lose sight of what biology is, what the biological system is doing, how the complexity is enabling it to maintain robustness in the process of oversimplifying these things, and I mean, kind of deceiving ourselves that it's much more simple than what it is. That complexity is something that is to be embraced. That is something that has to be accounted for at each and every step of whatever scientific discovery that we do, rather than just ignoring it and say that there is only one solution, it's all homogeneous. Synaptic plasticity is the only form of plasticity. Glial cells don't do anything. A neuron is a perceptron. These are all oversimplifications that don't fit with our current understanding of what the biological system that we have been studying has been telling us, basically, right? So each of these have to be accounted for in our analysis. Without that, it becomes extremely difficult to say that we are actually studying the system. If you just assume that this complexity doesn't exist, you are studying an imaginary machine which doesn't exist in reality. So therefore, if you're really interested in studying the brain, you will have to embrace the complexity. There is no other alternate to this, basically. Right? So that's that. I'll just stop. I have already overshot my time.
Great. Thank you very much. I think that's a very, expansive talk, touching a number of important themes and weaving them together. It sounds like a future review paper, because I think it is really quite important. We will open it up for questions. I think Maurizio has already raised his hand, so please unmute and ask your question. And anyone else who would like to do that, please raise your hand. And I think it's best if you ask your question directly if you're willing to unmute and do so. But Maurizio, take it away.
Thank you. Thank you, Steve. Thank you, Rishikesh. Very insightful. So I have I might have missed something when you were discussing the grid cell excuse me, study. So you have lots of heterogeneity over there, right? And the degree of heterogeneity disrupts the grid. You take into account resonance factors, and the resonance give robustness to the response. I'm not sure I understand. Of course, there might be a balance there, but in your framework, how would you argue that the network has to adapt, right? So the grid is going to change with respect to the context, right? There is some kind of tunability. So heterogeneity is something we want and we definitely have into place, but resonance itself, right? It's good to create the phenomena, but we don't want too much resonance, otherwise the network is not going to adapt. So in this framework, your interesting idea of the continuous learning, right? So I assume this being intersect with that aspect, but then my question is, how is your network able still to continue learning without forgetting? Because you still want to remember stuff.
So in that network, there is no adaptation, there is no learning. It's just a fixed structure with a specific connectivity, which is the Mexican hat connectivity that is connecting between these different neurons. It's just that the neurons, individual neurons, were replaced from the integrators that were present in the original paper to a resonator, which is more reflective of what is present in the entorhinal context, right? And to address the question of whether it is too much resonance, we just match the resonance frequency to the kind of frequency that the entorhinal cells have. There is no adaptation in that particular structure. The synaptic weights are fixed. It's just that so how this the grid pattern firing is constructed is you would have a virtual animal running in a two-dimensional arena, right? And the position of that animal, the velocity of it is fed into the neurons on this particular network. And that activity pattern propagates along this network as a bump. And that is what provides you this grid pattern firing rate, right? I mean, as the animal propagates, as the animal traverses in this arena, you would record the activity of one single neuron that is present in the network and ask questions about whether it is forming a pattern or not. So there is no adaptation, and that study we did not look at any form of continual learning or anything like that. The question was simply whether the presence of these different forms of heterogeneities would that hamper or would that enable grid pattern, activity patterns in this particular structure, or that was the fundamental question, and we found that it hampered. And then we asked, why is it hampering? And we found that it was because the lower frequency signals were kind of perturbed. And we asked if we suppress only the lower frequencies using quote unquote biologically realistic mechanisms, would this hampering be gone? And it turned out that happened. So the short answer is we did not have any continual learning in that particular section. Right?
Okay, thanks.
Thank you. Okay, well, Scott, Rich, you're up next.
Hey, thank you for that fantastic talk. So I'm very interested in specifically what you're talking about in terms of intrinsic heterogeneity. That's something that that we've looked at recently relative to sort of neuropathologies. I'm curious whether you have any sort of hypotheses as to how these heterogeneities might actually arise. Do you think that this is something that these might be accomplished by plasticity mechanisms, or developed throughout neurodevelopment? Do you think that this is just biological noise that just happens to have a functional role? What's your sort of hypotheses on that?
I'm inclined to say all of the above, with the lesser emphasis on quote unquote biological noise. Not that I disregard it or think of biological noise as something that is unimportant. We think of stochastic resonance as an important important robustness mechanism across all scales, and therefore, so my way of looking at it would be, there is a developmental component, of course, because it's not like all heterogeneities come through only because of postnatal or post-developmental, post-adult changes as a consequence of input activity that is coming from the external world. So there is that component. There is also activity-dependent plasticity, as I mentioned. There are several lines of evidence to suggest that ion channels also change, intrinsic excitability also change in response to behavioral patterns of activity, and therefore, and in the process of maintaining homeostasis in parallel. So you have all of this contributing to plasticity in ion channels as well. So as a consequence of that, you would have, say, for instance, for a given kind of activity pattern, one set of neurons might be responding more than the other. That brings about heterogeneities. And there is a certain noise component also which contributes to it. So, it's a nonlinear combination of all three is what I would think of where the origins of this intrinsic heterogeneity is. And of course, if there are pathological components that come into picture in terms of some kind of insult that comes from the external world, or sustained changes in activity patterns as a consequence of that initial insult, then that also could result in loss of heterogeneity, as Taufik's paper, I think. I don't know if you are an author on that paper.
Yeah.
Okay, that's very good. If you are an author on that paper, I thought that was a very fresh way of looking at heterogeneities and the link to epilepsy. Epilepsy has a lot of heterogeneities, and how all the neurons kind of start responding with high excitability, and therefore you have this how exactly it responds to synchrony, and the way that linked it to decorrelation and stuff like that. I like that overall idea. So that's something which I enjoyed reading that paper also. I read it when it was in bioRxiv. I have not read the new version of it.
Awesome, thank you. I was not fishing for the compliment, but I appreciate it nonetheless.
No. I already I mailed Taufik, so it's not a consequence of you fishing for it. It's something which is inherent, implicit.
Great. Okay, I think we have time for one last question from Francis. Francis, if you would like to ask it.
Yeah, actually, there's a question in the chat, which just give preference to that person.
Could you please provide your thoughts on pleiotropy and its oversimplification?
Oh, yes.
So that's the question. So degeneracy is the ability of different structural components to give exactly the same functional outcome. Pleiotropy, different people pronounce that differently. I pronounce it as pleiotropy. The pleiotropy is a scenario where the same structural component results in different functional outcomes in different contexts. So you can, or even the same. Let me give you an example. Say for instance, if you think of ion channels, so if you have HCN, for instance, it changes input resistance, it changes temporal summation, it changes resonance frequency, it changes oscillatory patterns, it changes so many different things. Basically, it's not like there is a one-to-one mapping between HCN will only affect resonance and nothing else. Right? So there is a one-to-many mapping between the structural components and the different functions that are present. So in degeneracy, we thought of we looked at different structural components giving you one function. Here you have one single component giving you various different structural functional outcomes, basically. Right? So that's what the combination of pleiotropy and degeneracy is, what gives you a many-to-many mapping between structural components and functional outcomes. So I mean, when we think of degeneracy, we think of it as a combination of pleiotropy and degeneracy together. But some people like to have it as two different things, because I mean, if you look at Edelman and Galley paper, they would mention that I mean, as a virtue of as a consequence of the presence of degeneracy itself, you would have the same component giving you different functional outcomes in different contexts. Therefore, we think of degeneracy as a combination of the two of them, and therefore we think of it as a many-to-many mapping between structural components and functional outcomes. But I mean, pleiotropy is precisely that, one-to-many mapping in terms of structure and function. So that is something that needs to be accounted for in analyzing things. You can't think of, I mean, sodium channels, they are going to be contributing only to the rising phase of action potential and nothing else. Delayed rectifier potassium channels, they are going to be only bringing down the action potential and nothing else. That's an oversimplification, attributing one function to it. Different contexts in different kinds of activity patterns, they would result in different kinds of function. So that is what pleiotropy means, and that's something that needs to be accounted for. I hope that answers that question.
Thanks.
Francis, do you want the last?
I guess
question.
Yeah, I
Keep it
mean,
quick.
I guess mine is a more general kind of question / comment to get your to hear your opinion about, since you've done many different types of examinations of oversimplifications in models and also doing experimental work. And so I completely agree with you about embracing the complexity in the biological system, as coming from Eve's lab, like, we have to care about the biological system. But then I guess my question / comment to you is like, there's always going to be more. And so from a strategic point of view of modeling, like how do you see these different aspects? So for example, in the different examples you provided in your talk, is it sort of more, so like the grid cell resonance part, including a certain amount of complexity in order to suggest functional aspects? Or is it more just showing the different complexities can matter? Like, so I guess I kind of want to get in your head a little bit to understand your thinking about these different things, because and I mean, it's just, yeah, we're all faced with this, right? Like we care about the biological system. We want to model the models to be helpful in terms of understanding it. But as it gets more complicated in different ways, it's harder for the models to be helpful to dissect out mechanisms. So yeah, so maybe just your comments or your thoughts about that.
Yes, it's definitely difficult to embrace the complexity in practice, of course. So I mean, the way I think of it is, I mean, we were able to do what we were able to do because in this particular scale, the molecular to cellular mapping scale, not, I mean, I don't think of it as a jump across scales. I always think of one scale jumps because, I mean, at each and every scale, you have degeneracy that comes into the picture. So, I mean, in this scale, because, I mean, the definition of complex system from the Edelman Galley perspective is that there are several different structurally distinct subsystems which are functionally segregated, and they all come together to which they call as functional integration to give a single function. Basically, in this scale, we knew what exactly those components were. We called them ion channels, and we knew that there were specific functions associated with them. We call them voltage gated gating or calcium dependent gating, and therefore there was a very functional specification. We did not define them in terms of amino acids. We did not define them in terms of quarks and leptons. We defined them as ion channels. Basically, there was that structure that was present, and we also knew what exactly the function was. I mean, in terms of cellular outcomes, action potentials, firing rate, resonance, and so on and so forth. And therefore, the idea of the ability to quantitatively assess if there could be several structural components which could come together to elicit an outcome became relatively easier in this scenario, even though the complexity is huge and there are so many different ways of doing it. But there are certain scales where that is still not possible. I mean, the best one is to jump from all the way from neurons to behavior. And there it becomes kind of, I mean, there is no, I mean, it's like thinking about ion channel to cellular functional map. So from the perspective of leptons and quarks or amino acids to, I mean, neuronal function. So in scenarios where the functional segregation has been identified, and these components, the subsystem have been clearly identified, and a specific function has been attributed to the emergent outcome, there this analysis can be carried out. I mean, in other places, it becomes very vague, basically. That's one thing. The second thing is I have always been against jumping across scales. I mean, say, for instance, jumping all the way from ion channels to behavior. What happens if I take out this ion channel? What happens to behavior, basically? Because at each and every scale, there is this considerable variability across different cells, different animals. And there is, I mean, deep degeneracy, as some people refer to it, at each and every scale. It becomes difficult to even go beyond one scale, basically. So I'm more of a fractal kind of I have more of a fractal kind of a view where at each and every scale you have fundamental components, and at each and every scale you have a functional outcome, and there are specific interaction which we call by different names. I mean, between amino acids we have this different kinds of interaction, and they have different kinds of folds of protein which gives us ion channels. Then different sets of ion channels contributing to specific functional outcomes. Different sets of neurons giving us, I mean, small networks which give us specific kind of functional outcomes at the network scale. So I think of at each and every scale, I mean, when we think of the network scale, we think of neurons as the fundamental component, and the network outcome as the functional outcome. And we think of neurons, we think of ion channels as the ion channels and other molecules that are present over there as the fundamental component, and the functional outcome at the cellular part to be the outcome, basically. So I don't think of jumping all the way from the molecular end to the behavioral end. So I think of at each and every scale, the analysis should be carried out at that particular scale. And I mean, a well-defined functionally segregated subsystem has to be identified at that particular scale, and a functional outcome has to be identified. And once that functional outcome is identified, there is no point in modeling a neuron as how exactly atoms interact with each other, or how exactly leptons and quarks interact with each other. It is sufficient to consider one scale below and take it forward. There is no need to, I mean, go all the way down there, basically. So I think of it as, I mean, every scale there is a fractal-like structure where we have different functional components. How they interact with each other is different. In ion channel end, you have voltage acting as the mediator. In the network scale, you have spikes acting as the mediator and synapses acting as a mediator. But I mean, I think of it as fractals, where the complexity is pretty much exactly the same at each and every scale, basically. And finally, I mean, I also do understand that it's not like we can think of it as a hierarchy as well, where it is molecule, cell, network, behavior, because as several people have shown, there is one change at one particular scale in parallel affect several different things across all scales. I mean, one can phosphorylate as many AMPA receptors in a dish. That is not going to contribute to memory. On the other hand, if you have that AMPA receptor present in a synapse, which actually alters the action potential outputs, which eventually goes and does something to the motor system that is present over there, then you have a behavioral memory. So that single phosphorylation event, or not single, that collective set of phosphorylation events associated with a bunch of AMPA receptors that were present across different neurons was able to do that only because of the context in which that particular AMPA receptor was present. So therefore, it at the same time, it's not like there's a jump of I mean events. It's at the same time affecting all the different scales. I do understand that. I mean, I don't know how to account for that in this overall scheme of where I think of it as fractals in each and every scale. I don't know the answer to that. I mean, okay. I don't know if I overall answered your question.
No, thank you very much. I mean, that was helpful, just getting the context, because I mean, it's something we all struggle with in terms of, you know, how to sort of, you know, get insights in different levels and simplifying, but not oversimplifying, as you put it.
Correct.
So thank you. I appreciate your comments.
Okay, we've hit the 90-minute mark, so I want to stop here, although there is one more question, but I know that people probably have to leave. So for anyone who has to leave, thank you very much for coming, and Dr. Narayanan, thank you very much for a very insightful talk. I would also like to thank the Crimble Computational Neuroscience for hosting this, and as well as the CPIN partner for hosting this. I think it was a great talk, a really good attendance. We lost a few people here during the question period, but I think it was also a very good question period. So for anyone who needs to leave, thank you very much for attending. We do have one more question though, Dr. Narayanan, if you can stick around, I will read it out.
I can read it.
Michelangelo, if you like it, that'd be great.
5 oversimplifications should imply an increase in the computational cost in the mathematical models. Any oversimplification for that matter, not only these 5, but any other things. That's one of the reasons why we do these simplifications, which is why I kind of made that as oversimplification. Now, in particular, when we want to study networks of neurons and glial cells, which correction to oversimplification could have a lower impact on the computational cost? If we had to account only one of these corrections to build a model, which one would you choose as non-negligible to stay as close as possible to the real system? Thank you very much. I mean, my answer is very simple. What's your question? So, I mean, this is the usual answer that I mean, the way we all think of computational neuroscience is from a utilitarian perspective. Right? At the end of the day, we are trying to address a specific question, right? We are not trying to, I mean, we are not in a race to get as close to reality as possible. That is something which we're really far away from, right? So we are not thinking of it as some kind of a race towards getting as close to the real system as possible. We are asking, okay, this is my specific problem. This is my biological problem that I'm trying to address. And I'm interested in building this computational model with this falsifiable hypothesis towards asking whether this is possible or not, or can it give me some predictions, basically. So depending upon your question, if your question is with reference to the interaction between glial cells and neurons, then obviously you will have to account for what exactly glial cells can do and how they interact with neurons. If your questions are with reference to, I mean, the presence of dendrites, the presence of ion channels, and so on and so forth, then it is important to, account for the presence of active dendrites. There is no point in introducing one additional component just for the heck of it. That's not going to help you, because you want to make sure that you don't, as Francis also mentioned, you want to simplify but not too much, right? And it's the right level of detail that you want to use for your particular model. And that entirely depends upon what your question is. The question is always the master. The rest of it is dictated by what that question is. If you want, if you want, say for instance, for a given ion channel, you can have a Hodgkin and Huxley type model. You can have a 75 state Markovian model for that. So which one do you choose? Which one do you choose? It depends upon the question again. If your question involves the different states of the ion channels, and if you want to look at transitions and stuff like that, you want a Markovian transitioning channel model rather than taking a Hodgkin and Huxley model, which assumes independence of, say, activation and inactivation, basically. Right? So things like that come into picture, and at the end of the day, my only answer to all these questions as to what level of model is correct, what details should I include, what details should I omit, the answer is very, very simple. What is your question? The question basically determines the level of details that you want to put in the model. There is no point in putting additional parameter. An additional parameter is always dangerous. If you don't want that model, don't want that parameter, do not put it. It's just going to make your life much more difficult than what it would be otherwise. That would be
that's good advice. Depends on the question, for sure. Okay, with that, I think let's all thank our speaker for today. I know it's getting late for you, so thank you very much for sticking it out, and we really appreciate it. Thank you again.
Yeah, thank you very much.
Thank you so much for having me there. Thank you so much. Thanks.
All right. Okay. Bye-bye.
Thank you.
Bye all.
Bye. Thank you.